Hi everyone, thank you for being here today. I would like to also thank the DW committee for this opportunity, and I have no disclosures. So the microbiome, or the human microbiome, is made up of several types of microorganisms from different kingdoms, including bacteria, archaea, fungi, viruses, and protozoa. And although representing less than 1% of the microbes, archaea are important but overlooked part of the microbiome. Many of these archaea produce methane, and they live in the human gastrointestinal tract. One key species here is Methanobrevibacterium. While bacteria are the most studied component of the gut microbiome, archaea species are increasingly recognized for their roles in human health and disease. And research shows, largely based on stool analysis, that archaea can influence not only the resident gut microbes, but also several host metabolism process and energy balance, and especially in the small bowel, where digestion, absorption, and metabolic hormone signaling occur. And as part of this process, archaea consume large amounts of hydrogen, which increases energy yield from the diet. But how and why is hydrogen consumed? Hydrogen is primarily consumed during methanogenesis, which is the process by which certain archaea produce methane. And methane itself can affect human physiological pathways and influence metabolic outcomes. So these figures represent a conceptual framework linking methane to human physiological effects and potential health outcomes. Methane is shown here as being produced primarily in the gut by methanogenic archaea. And moving downwards, methane and methanogens interact direct or indirectly with several key systemic pathways, including the gut microbiota, the enteric nervous systems, the intestinal barrier immune response, and other metabolic pathways related to energy harvesting and thus possibly metabolic hormonal signaling. This interaction leads then to physiological effects. This includes changes in gut fermentation and gas dynamics, changes in gastrointestinal motility, modulation of intestinal barrier immune function, energy balance, among other possible effects. Further downstream, these changes may contribute to systemic effects and outcomes related to obesity, overweight, shifts in inflammation and metabolism. And the interrogated color here at the end is meant to show that these processes are connected rather than isolated. So methane production is not just a biological process, it actually has clinical significance. So in weight disorders, specifically in underweight and malnutrition, Methanobrevibacter smithii appear to be enriched in the bowel of these subjects. It's well accepted that might represent an adaptive microbial response to limited nutrient availability and is aimed to improve energy harvesting from the diet. In obesity, increased levels of methanogens and methane in the breath are also associated with greater BMI and body fat, as highlighted in these two studies. And in this case, it might be adaptation from the diet. Previously, we showed that Methanobrevibacter smithii biomass can be manipulated by changes in diet. In this study, a high-fat diet rats stimulated the propagation of this archaea throughout the gut. But more importantly, rats that gained the most weight were the ones where Methanobrevibacter smithii was detected in more than one GI segment. And those that gained the least weight, even on a high-fat diet, were the ones where Methanobrevibacter smithii could not be detected in any GI segment. Previously, we also showed and demonstrated that methane is a gas transmitter that slows intestinal transit by infusing this gas in the animals. So our proposed hypothesis here is that when Methanobrevibacter smithii produce methane, besides slowing intestinal transit, a large amount of hydrogen is consumed, enabling for a more complete oxidation of substrate and ultimately improving energy harvest from the diet. In scenarios where energy is needed more, such as in cases of underweight and malnutrition, slowing the gut and harvesting more energy from food benefits the host. In cases where more energy harvesting is not needed, such as in subjects with obesity, increases in methanogens likely due to their diet lead to an undesirable outcome that might also worsen glucose and insulin homeostasis. Now, considering what we know about these scenarios and the importance of the small intestine in diet, nutrition, and metabolic process, we aim with our study to examine the absolute abundance of methanogens in duodenal aspirates of subjects across a spectrum of. For that, we select subjects from the reimagined cohort at Cedars-Sinai. Currently, we have the largest collection of small bowel aspirates for microbiome studies in the United States, along with blood samples, biopsies, and medical records. We recruited subjects undergoing upper endoscopy without colon prep, and we collect duodenal aspirates using a custom double lumen catheter to avoid cross-contamination with other sites, including saliva. We extracted the DNA from duodenal aspirates, we added internal controls, and performed quantitative whole genome sequencing for metagenomic analysis. And this is the demographics of the BMI cohorts utilizing this study. The first piece of results that I want to share with you is that the duodenal microbial diversity, including bacteria and archaea, and based on quantitative data, was similar across BMI groups. Here, I also show the distribution of subjects with small bowel bacterial overgrowth, or SIBO, represented by the pink circles. SIBO is one of the most well-known microbiome-related in the small bowel, in which an excessive number of gram-negative bacteria with a high capacity for producing hydrogen are detected. And let's keep that in mind. But we did not observe differences in SIBO prevalence across different BMI classes. In the next results, I want to share the archaeon profiling in each group, represented here at phylum level. So this is only to show you that the phylum Methanobacteriota, the blue color here, from which many methanogens belong, is the most abundant phylum in the small bowel. But if you want to know more about the specific Archaea species, Dr. Villanova Milan from our group will present two interesting posters on Tuesday. One showing the full characterization of this kingdom and duodenum samples, and one showing a little more about the gas dynamics in the small bowel, including methane. But let's take a look at the specific methanogens level in our study. So when we analyze the absolute abundance of specific methanogenic archaea, the absolute abundance of several methanogens were altered in subjects with underweight, including higher absolute abundance of Methanobrevibacter smithii when compared to subjects with a healthy weight. And similarly, the absolute abundance of Methanobrevibacter smithii was also higher in subjects with obesity when compared to subjects with a healthy weight. And no differences we observe in subjects with overweight. The visualization of these results is even more revealing. So, as I just told you, subjects with overweight have a similar absolute abundance of Methanobrevibacter smithii in the duodenum when compared to healthy weight. On the other hand, the absolute abundance of M. smithii in underweight and obesity was higher when compared to healthy weight, suggesting a U-shaped pattern related to BMI extremes. When we check breath levels of methane from subjects with a duodenal microbiome with increased capability of producing hydrogen, such as in SIBO, subjects with overweight were also similar to healthy weight, while methane appeared to be higher in subjects with underweight and obesity, suggesting a similar pattern here too. We had some in our study. We did not take into consideration for this analysis diet and macronutrients, including the fat intake, and the absence of a full lactulose breath test data. So in conclusion, the absolute abundance of duodenal methanogens is associated with extremes of BMI, and the absolute abundance of Methanobrevibacter smithii follows a U-shaped pattern across BMI categories, and it's higher in both subjects with underweight and obesity. Breath levels of methane suggest a similar U-shaped pattern. And in subjects with obesity, the high absolute abundance of methanogens might be actually a maladaptive response. Thank you for your attention. I would like to thank the MAST team Without them, I couldn't be here presenting these results today. I would like to thank our participants as well, and now I'm open for questions. Thank you.